Welcome to the fascinating world of genetics. Have you ever wondered why you might have your mother's eyes but your father's smile? Or how two brown-eyed parents can suddenly have a blue-eyed child? Today, we are going to uncover the fundamental rules that govern these biological mysteries. We will explore Mendel's laws of inheritance, specifically the principle of segregation and the principle of independent assortment. By the end of this lecture, you will understand exactly how traits are passed from one generation to the next, and you will even be able to predict the genetic outcomes of various pairings. To begin our journey, let us look at Mendel's first law, known as the principle of segregation. Why does this matter? Before this principle was understood, people believed traits simply blended together, like mixing paint. This law proved that traits are actually passed on as distinct, discrete units. Imagine you have two different recipe cards for cookies, but you can only give one to a friend. In genetics, your body does something very similar. You carry two genetic recipe cards called alleles for every trait. However, during the formation of gametes, which are the sperm and egg cells, these two alleles separate from each other. Because of this segregation, each gamete receives only one allele. When fertilization occurs, the offspring gets one allele from each parent, restoring the pair. Now that we understand that alleles separate, let us look at how we can predict the outcomes of this separation mathematically. This formula comes directly from the rules of independent probability. The variable p of g represents the total probability of an offspring inheriting a specific genotype, which is their genetic makeup for a trait. The symbol p1 represents the probability of inheriting the first specific allele from parent 1. The symbol p2 represents the probability of inheriting the second specific allele from parent 2. We multiply these two probabilities together because these are independent events happening at the exact same time, much like flipping two coins and getting heads on both. If the chance of getting an allele from one parent increases, the overall probability of that genotype increases proportionally. Before we move on, let us see this formula in action with a concrete example. We are given two parents who are both heterozygous for brown eyes, meaning they each carry one dominant brown allele and one recessive blue allele. We are asked to find the exact chance of their child having blue eyes. To do this, the child must inherit the recessive allele from both parents. We chose our multiplication formula because these are independent events. The mother has a 1 in 2 chance of passing on her blue allele, so PM equals 1/2. The father also has a 1 in 2 chance, so PF equals 1/2. Multiplying these together gives us 1/4. Physically, this means there is a 25% chance in every pregnancy that the child will have blue eyes, showing how hidden traits can suddenly appear. Let us apply this to another practical scenario called a test cross. A farmer has a tall pea plant, but does not know if it is hiding a recessive short trait. To find out, he crosses it with a short pea plant, which we know is strictly recessive. We want to find the proportion of short offspring if the tall plant is indeed hiding the recessive allele. We use our probability formula again. If the tall plant is heterozygous, its chance of passing the recessive allele is 1/2. The short plant can only pass the recessive allele, so its probability is 1. Multiplying 1/2 by 1 gives us a final answer of 1/2. This means 50% of the offspring will be short. If the farmer sees this result, he knows for sure the original plant was hiding that recessive trait. Now that we have calculated these probabilities, let us look at a visual representation of what is happening. This is a Punnett square for a monohybrid cross. Along the top, you see the two alleles from one parent, labeled capital B and lowercase b. Along the left side, you see the alleles from the other parent, also capital B and lowercase b. The 4 boxes in the middle represent the possible genetic combinations for their offspring. Notice the bottom right box, labeled lowercase b, which is highlighted in blue. This visually confirms our earlier calculation. There is exactly 1 out of 4 boxes that results in the double recessive trait. This diagram perfectly illustrates how the alleles segregate and then recombine during fertilization. Having mastered how single traits are passed down, this brings us to Mendel's second law, the principle of independent assortment. Why is this important? Without this principle, traits would always be inherited together in rigid blocks, severely limiting variation. This law states that the alleles of 2 or more different genes get sorted into gametes completely independently of one another. Imagine packing for a trip. Choosing a red shirt does not force you to choose blue pants. Similarly, inheriting a specific hair color does not restrict which eye color you inherit. This happens during meiosis, when homologous chromosomes line up randomly at the cell equator. It is crucial to note that this applies specifically to genes located on different chromosomes or those very far apart on the same chromosome. Building on that random sorting, let us explore why independent assortment is considered the biological engine of genetic diversity. This concept is vital because genetic diversity is what allows a species to survive. If a new disease or environmental change occurs, a diverse population is much more likely to have individuals with the right combination of traits to survive. Because chromosomes line up randomly, a massive variety of genetic combinations is created in the resulting gametes. This is the exact reason why you and your siblings can look so incredibly different, even though you share the same parents. Furthermore, agricultural scientists and animal breeders rely heavily on this principle. It allows them to mix and match desirable traits, like breeding a cow that produces high milk volumes with one that is highly resistant to heat. Now that we understand the concept of independent assortment, let us look at its mathematical form. This formula extends our previous logic to multiple traits. The variable p of a, b represents the probability of 2 independent genetic events happening at the exact same time, such as inheriting a specific seed color and a specific seed shape. The symbol P of A is the probability of the first event occurring, and P of B is the probability of the second event occurring. Because the principle of independent assortment tells us these traits do not affect each other, we can simply multiply their individual probabilities together. If the probability of either individual trait changes, the combined probability of inheriting both traits will change as a direct mathematical product of the two. Let us see this new formula in practice by predicting 2 traits simultaneously. We are given 2 pea plants that are heterozygous for both seed color and seed shape. Yellow and round are dominant, while green and wrinkled are recessive. We are asked to find the exact probability of an offspring having both green and wrinkled seeds. Because these traits assort independently, we chose to treat this complex cross as 2 separate simple crosses. First, the chance of getting green seeds from 2 heterozygous parents is 1/4, so PY equals 1/4. Second, the chance of getting wrinkled seeds is also 1/4, so PW equals 1/4. Multiplying these together gives us a final answer of 1/16. Physically, this means out of every 16 offspring, we expect only 1 to have both recessive traits. Now that we have successfully calculated 2 traits, let us scale up and add a 3rd trait to our genetic puzzle. We are now crossing 2 plants that are heterozygous for seed color, seed shape, and plant height. We want to find the probability of an offspring being completely homozygous recessive for all three traits: short, green, and wrinkled. Drawing a grid for this would require 64 boxes and be incredibly confusing. Instead, we use our independent assortment formula. We already know the probability for green seeds is 1/4 and wrinkled seeds is 1/4. The probability for short height from heterozygous parents is also 1/4. We multiply 1/4 by 1/4 by 1/4 to get a final answer of 1 in 64. This shows how rare it is to inherit all three recessive traits at once and demonstrates the mathematical beauty of breaking complex problems into simple pieces. To truly grasp how we got those probabilities, let us look at the cellular level. This diagram shows exactly how independent assortment happens during meiosis. You're looking at two possible ways chromosomes can align at the cell equator, labeled alignment 1 and alignment 2. The cell has two pairs of chromosomes, distinguished by size and color. In alignment 1 on the left, the red and yellow chromosomes line up on one side, leading to gametes that carry that specific combination. But look at alignment 2 on the right. Here, the red chromosome lines up with the green one instead. Both alignments are equally likely to happen. This visual perfectly captures the physical mechanism of Mendel's second law. It shows that inheriting the red chromosome does not dictate whether you get the yellow or the green one, creating 4 genetically distinct gametes. We have covered a tremendous amount of ground today, so let us summarize the most important ideas. First, we learned the principle of segregation, which guarantees that the two alleles for any trait separate during gamete formation. This ensures that a child inherits exactly one genetic instruction from each parent, keeping our genetic material balanced. Second, we explored the principle of independent assortment, which tells us that different traits are inherited independently of one another. This random mixing is the ultimate engine of genetic variation, ensuring no two individuals are exactly alike. Finally, we saw how these biological principles translate into mathematics, allowing us to predict complex genetic outcomes using simple probability. You now have the foundational tools to understand how life passes on its biological blueprints. Excellent work today.